Talks, your daily dose of health and medical news. I'm Mr. Zaman, and here's what I bring to you all from the world of medicine. How statin therapy can yield lifelong benefits. Stopping statin treatment early could substantially reduce lifetime protection against heart disease, since a large share of the benefit occurs later in life. That's the finding of a modeling study presented at ESC Congress 2022. Statins are the most commonly used lipid-modifying drug. It is estimated that in 2018, they were taken by more than 145 million people. However, delayed treatment initiation and poor adherence are common. Exaggerated claims about side effects of statins may also be responsible for underuse among individuals at increased risk of cardiovascular events. There is some uncertainty about when to start and how long to persist with statin therapy to optimize the effects. This study estimated the accumulation of benefit with statins according to age at therapy initiation using a microsimulation model that was developed using data on 118,000 participants of large international statin trials from the Cholesterol Treatment Trials (CTT) collaboration and 5 lakh individuals in the UK Biobank population cohort. The researchers found that a large part of quality adjusted life years or QALYs gained with statin therapy accrued later in life. The higher the participants' 10-year cardiovascular risk, the larger and earlier the statin benefit accrued. Compared with lifelong statins, stopping therapy at 80 years of age erased a large share of the potential benefit, especially for people with relatively low cardiovascular risk. Consequences of smoking. Smokers have weaker hearts than non-smokers, according to research presented at ESC Congress 2022. This study explored whether smoking was related to changes in the structure and function of the heart in people without cardiovascular disease and the effects of changing smoking habits. The study used data from the 5th Copenhagen City Heart Study, which investigated cardiovascular risk factors and diseases in the general population. A total of 3,874 participants aged 20 to 99 years without heart disease were enrolled. A self-administered questionnaire was used to obtain information on smoking history and to estimate pack years, which is the number of cigarettes smoked through life. One pack year is defined as 20 cigarettes smoked every day for 1 year. The researchers compared the echocardiography measures of current smokers versus never smokers after adjusting for age, sex, body mass index, hypertension, high cholesterol, diabetes, and lung function. Compared to never smokers, current smokers had thicker, weaker, and heavier hearts. Increasing pack years was associated with pumping less blood. The good news, they said, is that some of the damage is reversible by giving up. The new blood test found by researchers at the National Institute of Health to evaluate the severity of pulmonary arterial hypertension. This new blood test measures DNA fragments shed by damaged cells. Researchers found that these fragments, called cell-free DNA, were elevated in the blood of patients with PAH and increases with disease severity. Patients with PAH have a high death rate, and the condition mostly affects women. Despite treatment advances, it currently has no cure. Current tests used to monitor PAH severity rely on established risk prediction scores based on clinical symptoms and on the use of an invasive catheter to measure pressure in the lungs. Doctors sometimes use echocardiography or heart imaging to measure pressures in the heart as an indirect measurement of lung pressure, but these tests tend to lack reliability and sensitivity both. In the current study, the research team analyzed cell-free DNA from blood samples taken from 209 adult patients, predominantly women. The researchers compared the results to cell-free DNA measured from a control group of 48 healthy volunteers without PAH at the NIH Clinical Center. They found that cell-free DNA was elevated in patients with PAH and also found that cell-free DNA concentrations increased in proportion to the severity of the disease. Patients with the highest level of cell-free DNA had a 3.8 times greater risk of either death or a need for lung transplantation compared to those with the lowest level of cell-free DNA, the researchers said. Further analysis of cell-free DNA samples revealed that multiple tissue types, including the heart, blood vessels, fat tissue, and inflammatory cells circulating in the blood, were affected by PAH. This knowledge may lead to new drug interventions for PAH, whose current treatment options may slow but not halt or reverse disease progression. of synthetic mouse embryo from stem cells which has brain and heartbeats. The results are the culmination of more than a decade of research, and they could help researchers understand why some embryos fail while others go on to develop into a fetus as part of a healthy pregnancy. Additionally, the results could be used to guide repair and development of synthetic human organs for transplantation. The embryo model was developed without eggs or sperm. Instead, the researchers mimicked natural processes in the lab by guiding the 3 types of stem cells found in early mammalian development to the point where they start interacting. By inducing the expression of a particular set of genes and establishing a unique environment for their interactions, the researchers were able to get the stem cells to talk to each other. The stem cells self-organize into structures that progress through the successive developmental stages until the synthetic embryos had beating hearts and the foundations for a brain, as well as the yolk sac where the embryo develops and from which it receives nutrients in its first weeks. This is the most advanced stage of development achieved to date in a stem cell derived model. A major advance in this study is the ability to generate the entire brain, in particular the anterior region, which has been a holy grail in the development of synthetic embryos. The researchers found that the extra embryonic cells signal to embryonic cells through chemical signals, but also through touch, guiding the embryo's development. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.